Yeah, it takes very little to cool it down. But we hit 3.
And it's climbing, 3.5.
Nice. All right, we're just going to hold here for a moment. When you have the arm live and the vehicle and everything, like this is different because we're actually like set down on something. But if you're just towed in onto a cliff and you're using the arm, you have the the arm can run away. That's a known. We haven't had issues with that recently, but that's a known feature.
Feature.
Yeah, feature. At which point you'll really just kind of either kill power or kill the valve. And so there are things to be aware of, or like you can, if you just have literally 2 inches worth of the front porch on a ledge, that can slip. That octopus looks like it's got some damage on it.
Really scarred, yeah.
Yeah.
Wow.
Aaron, push all the way in.
Yeah, that's unusual.
The eggs have eyes.
We saw earlier this year in the Pacific Rim Islands a Grimpatoothless octopus that looked like it had like a head injury, like full slice across the top that had actually completely healed. Wow.
There's a good
like
shot
that,
of it.
yeah.
So, yeah, I've never seen anything like that. We usually when they go senescent, and you know, this is in the latter stages because we can see the developed embryos, they get kind of more marbly, a little more wrinkly and a little more shriveled, but I don't know if that's senescence or, yeah, just an injury. I wonder how that would have happened.
Wow.
We collected a male back in August, and it was fully senescent. I mean, it was barely alive, and it was missing half of an arm, another arm, and parts are essentially falling off of it. It was kind of dissolving, even though it was still alive, but this seems different.
Do we have any sense about
4.
who would prey on these species?
That's a good question. Yeah, I don't know.
Moving a bit.
It is. It does seem interesting.
I was thinking kind of in the same
sitting
spot
here
though,
so
'cause
vulnerable.
it's just
Yeah.
rotating
You know?
in the jaws, but
And you'd think if it was major predation, they wouldn't be out in these numbers. So most likely they're fine.
Do we have a species ID on the whelks that we've been seeing? There's one on the right-hand side.
Yeah, so we collected those and got them sequenced for my colleague at Mbari, and it's new to GenBank, and no one at Mbari knows what it is. So at least it's not common in the deep sea in Monterey Bay National Marine Sanctuary, and the photographs have been sent off to a couple of whelk experts in Italy, but it is quite possible it is a new species of whelk.
the oxygen saturation just drops.
And I wonder if it's a species that has, or that is, you know, typically found around seep sites.
Yeah, exactly. They certainly seem to be the trash guys here. They do a lot of grazing, eating of egg cases, and
Trash guys.
things of that nature.
The flow is low enough that it just takes a
Hey,
long
somebody's
time
gotta do
to
it.
really
Yeah.
reach the
Those
sensor.
are free
Yeah, and
nutrients.
heat up
Exactly.
the plastic.
Help
and
recycle.
everything else, yeah.
Because it comes down with a large volume of ambient seawater.
Have you all been seeing these hydroids a lot in the top right corner
Yes.
in association too?
And yeah, we want to, basically, even though we have some of these animals, we want to collect multiple replicates, maybe even for voucher specimens or later analysis. So we're going to try and collect one of those large anemones that could be attached to a smaller rock and easy to transport.
Yeah,
A rock
I'd come
with-
down. That's a tether.
A rock
tuck.
with those little
Pull wide.
anemones, those little actinaria on them, certainly whelks.
The
Perfect.
ship
We
drifting.
want to get
or-
an assemblage of
Nope.
these, this
Oh, maybe there's
seep
a current
species, if you
change.
will, the seep
Could be.
community.
And Chad, what's a voucher specimen?
Do you want me to move Argus closer to you?
I mean,
Yeah,
technically,
just
it's
a
a
little bit. Give
specimen
George
that
something to do.
acts as the
Alright.
original.
I'll just go
that
south.
the species is based on in
15
terms of the
meters.
description, and one that
Yeah.
scientists can keep going back to
Bridge
for
net.
that original description. So it may not be the correct terminology, but certainly for
One
OET's
step,
collection,
1.5
it
meters,
could be
bearing
considered
south.
a voucher for them, and for scientists to check out at a later date.
Yeah, we're in a different position than we were. That could be part of it.
you're right, I suspect just current.
So we have a group of 3rd grade citizen scientists tuning in from Forks, Washington, wondering what in the venting fluid helps make a more diverse animal count or diversity of species in this area.
That's exactly
Well,
what
if
we're
they
trying to figure out.
if they
right?
know the answer to that question, we'll hire them.
Exactly. Keep tuning in. That's why we're here too.
No, see, that's a great question, and that's one of the many questions we have. That's why we're here. This was just discovered a year ago, and we're just beginning to look at the tip of the iceberg here. We really
you
have
said you
no
want
idea.
to try the darker ones in the
yeah.
bottom?
So we peaked at 5.5.
5.5 there? Okay.
Yeah.
Yeah. I'll let it cool down.
And the oxygen concentration did dip, as expected.
Oh, she's got an arm through one of the bolt holes.
So it dipped from essentially an ambient of 27% saturation down to about 18%. So pretty significant drop.
Video, let's push in. We'll find a spot in the meantime. Hold that. Let's see.
But Chad, it's kind of a
Yeah, some
feature of
shimmer
these
there.
patchy, unique
I don't
Yeah,
see much
behind
shimmer
habitats
here.
that
Yeah,
show
I
up on the
agree.
deep sea floor, like
think
whale
the shimmer's
falls
behind
and these
that
kinds of
octopus
outcrops, where you
there.
find a lot of unusual organisms. It's interesting.
Yeah, the seamount as a whole is an oasis for deep sea corals and sponges. Could act as a nursery to reseed other rocky areas.
Right.
Like you said, whale falls are a boon to
Yeah.
deep sea fauna because they receive so little food relative to their shallow water counterparts. And
Exactly.
something like this, although we don't know what is beneficial, something is beneficial here,
Right.
bringing all of these animals together.
Yep.
There could be some symbiotic relationships going on. There could be bacterial help. We don't know the
Right.
answer to those questions yet. There could
I
be.
feel like that's
Hey,
exactly
Paul,
where I
that-
was.
There's a number of
Paul, that
things that could
move's
be going on here.
complete.
Yep.
I expect you might have to
Wait,
change
15
your heading
minutes.
to port. And then you can see kind of the depression we're in Argus Meso. We're getting closer to a rise, but should be okay.
And what's really interesting, I think, is working for
Video,
Monterey
let's
Bay National
control
Marine
Y.
Sanctuary. This place was added in 2008 based
Yeah, that's
upon
where we
the
were
deep sea
before.
corals and sponges we found on Davidson Seamount in the previous decade. And a year ago, we were exploring the flanks of the seamount here where no one's ever been before, looking for corals and sponges, and we ran into this. And now that we've seen developing embryos and hatching embryos, I think it's reasonable to assume this is a fairly successful population and could be acting as a nursery to reseed the surrounding seafloor with thousands and thousands of hatchlings every year.
Absolutely.
And so now the sanctuary is inadvertently but serendipitously protected an octopus nursery. So
Right.
that kind of speaks to the reason why we want to set aside special areas, even if we don't fully understand them, because that's quite often the case.
All right, let's,
it's-
Chad,
it's
there's not a
often
lot
a-
of flow in the darker rock. So if we zoom in, video,
Yep. Let's get this reading here.
hold that. Yeah, so we don't have very much flow.
Oh, there's not much flow here.
Oh, there's some behind.
There's a little
there.
bit behind that octopus, but we were previously right in that
we were
spot.
just a few
Yeah,
inches
So-
away.
That's right.
Okay, well that's fine. Let's go ahead and put the sensor away. We just want to
Okay.
get a couple of readings here and there, looking at the big picture of this 30+ hour dive.
All right,
We want to start
full
making
wide.
collections and eventually start our transects, because one of the goals of this dive is to find more octopus and see how big and how big this population is on this knoll. So
And Chad, it's often a feature of the reproductive strategies of organisms that inhabit unique, patchy environments like this, is that they brood their young. Because if they're restricted to a certain kind of habitat and the adult has found that, then they want to keep their young close to that habitat. So they brood until they can hatch out and then find their own place in that unique patchy habitat. Instead of spewing their gametes, say, in the ocean and hoping that they will be fertilized and then drift through the ocean and find another similar spot.
Yeah, well, we just don't know where these babies go, these
Right.
hatchlings go.
Right.
I mean, if they do indeed go into the water column, they could be transported very far away.
That's true.
To repopulate other areas.
Yep.
Or maybe they're active swimmers and they come back down to the benthos and
Yep.
grow
So
up
what's
there.
on our sample list?
Okay, so almost everything you see here.
All right. Because we have that little anemone on a rock right in front of us.
Yeah.
Video zoom in.
That would be a good place to start, if it's small enough.
That one.
Yeah, if that's
Yeah, I think I can get that rock.
great. That'd be fantastic.
Hopefully the anemone does not detach. All right, full wide.
I got it too.
Ooh, you want to switch back over to the tension?
1549.
Mm-hmm.
Peak 14.5. Okay, so we're still trending in the right spot. We're going to get more and more of those heaves. Video, let's push in.
Do we still like this delta? It's bouncing between 21 and 24.
As you come closer, we'll come up, but you haven't come that much closer yet. Let's lower our grip force. Let's do like 6.
It looks like we've finished our move, right?
You have finished your move, however, you have 3,000 meters of cable,
Yeah.
and you're dangling at the end of it.
Yeah, I think you moved a bit, but I don't know if you'll see all the lasting effects for a while. I can bump more, but I think for now, I don't think it'll get tugged.
No, I think we'll be okay.
Hard to say what size the rock. Oh, it's a little one. Teeny.
I know, but I'm
It's closing up.
trying to grab the rock, not the anemone.
Yeah.
You let me in there, I promise I'll be out shortly.
And don't hang on.
All right, we're gonna. Kind of steal that rock from you. And then whole vehicle, why you gotta move on me.
That was my front porch.
Nice.
Yeah, excellent.
All right, video, let's come full wide.
All right.
Herc, if we could put this in one of the small starboard bio boxes, that would be great.
Sure. Yeah, we should be good for that.
Now, Chad, are we seeing
Do you
these?
want to zoom or anything? Or are you happy with it, Jamie?
I think we're good.
Okay.
Are we seeing these same types of organisms in the different areas? So we've got the anemones, the shrimp, the whelks.
So again, I do my same
seeing
rotate
similar
around, right?
amongst all the different patches.
Well,
Do you want the
similar,
starboard
but I was
salvo?
describing
I would
earlier.
love
There's
the
been one
starboard
patch.
salvo, but you're gonna have to change the camera
Oh, change
on
the cameras. There was one patch where all the whelks seem to be replaced by sea stars.
Ah.
cameras.
So there's definitely some
starboard bio box,
variability to
port
these
rail,
seams,
perfect.
but I'd say, yeah, predominantly it's fairly similar
Okay.
from pool
Still
to pool.
have-
But That's kind of what I'm
Jamie,
excited
we're
about
good
later
on any
today
of
when
these?
we're running transects.
Yes.
To
Yeah,
see
it's
kind of
just
the
kind of see
variability
the
in the pools.
variability, yeah.
Because again, we've really barely seen
Let me
more
know when
than
you're ready for the
maybe a couple dozen
Couple
pools
Ready
spots,
for
here.
the
yeah.
box.
sample tray out. And we have no other samples in here yet.
Correct.
We do have a sample in E, which is one of the large ones, but it's a rock.
Okay, we're going to go to D.
Okay.
Just because that one can be hard for corals.
Do you want me to turn off one of those lights?
Yeah, go ahead and kill port starboard. That one. Sorry. Starboard port.
Got it.
Port starboard. Can't listen to me.
Ever do?
I know, right? So again, this is like a,
Ooh.
that
Did it
went,
go in?
Nope, it went to the outside.
Are you sure?
Yeah,
I don't know.
pretty sure it went outside.
Can't tell.
We can close the box and see if we can see.
We probably won't be able to
Yeah.
see,
Let's
but
get another one. Put it on top.
let's do
2 anemones. I don't think we'll be able to see
I'm
it.
pretty confident that went out of the box.
Okay.
Unless anybody says very specifically they saw it, we'll grab another one. We can leave the box open.
Okay.
Yeah. Let's go to normal forward.
Hopefully we can get one the same that can also fit in D. We can just double it.
Jamie, are you okay with all of this?
Yes.
Okay.
Oh, we have a second before the new sample. Is Argus still in a good position?
Yeah. All right, video, let's push in. Hold that. Good small rock with an anemone.
That one? That one with the lasers on?
Was the
Maybe.
one in the bottom left? Not, it's not in the picture anymore. That one, now it's in the middle with the lasers on it.
Maybe. I was also thinking about that one. All right, Aaron, let's come full wide. I'm just going to see if anybody's attached to a small rock. I think that might actually be a winner. Aaron, let's push in. Hold that.
I think that's on a big one, if I'm not mistaken.
Yeah, you win.
I can't tell. It might be a small one on top. Yeah, that might be just a little one.
I think the little one I moved is not the one that it's on.
Yeah, that's what I mean. It's not one part. It's not one big thing. Yeah.
I know, but I gotta get the rocket, not the anemone.
Nice grab. It's a nice rock.
So
All
Greg,
right, Jamie,
Greg,
we're
is
doing the
this-
same thing.
This is practice for
Yep.
doing laparoscopic surgery.
Yep.
Yeah, let's, we'll get a tight zoom of some of this rock because I want to see what else is growing on the side there.
All right, Aaron, give it what you got.
Wow, wow, look at that. A reddish, I bet that's,
Oh.
I don't know, bacteria on the rocks.
It's like altered rock,
Yeah.
thermally altered, chemically altered.
Yeah.
For what it's worth, Greg, you might only have 2 of those claws on it.
Yep.
Keep your hand up a little bit.
That happens frequently.
Thanks.
Great look at the texture on this. Talk to the anemone there.
Yeah.
All right, I'm going to go stow it.
Yeah, go ahead and stow
Okay,
it.
full line. Let's play this game again.
So, great,
Ooh.
Ooh.
I was getting stuck on the puck. There you
Yeah.
go.
great combo sample here, a little rock sample, and then an anemone. 2 checks.
Yeah, let's stay on the sample, that way we know. Okay.
Starboard salvo.
Yep.
So if you're watching on the quad view, check that out on the lower left.
Is that better?
You can see sort of reaching back around the right shoulder of the ROV. Think about putting your arm kind of behind your back and depositing those samples into the bio box on the starboard side.
Oh, octopus, you're in just the wrong spot.
It's our little parrot for the day.
Yeah.
For real. Chad, can you talk for a minute about why you're interested in the geologic samples or in the sample with the anemone on it? So,
Well, the
what are you
geologic
hoping to learn?
sample is just kind of a bonus.
Okay.
It's just hard to scrape off
All
these
right.
anemones and keep in one
Jamie,
piece.
I'm going to drop it.
Looks good.
So we're taking a
Oh,
2-for-1 here. But we
Now
have some people at
confirming
USGS that are
009.
interested in
009
rock samples
comes
Go ahead
anyway.
and close
Thank
up
you.
our box.
And what type of things are you hoping to learn from getting an anemone back?
We do know that this exists kind of cosmopolitan-like here, but we can do some isotopic studies to see how it differs from other animals that are not within the seep community as well.
And that would tell maybe different food source.
Correct. Thanks, Dan, yeah.
Fantastic.
Normal.
So Amity had a good idea.
Okay.
Back in that spot where you just got that anemone, there was
Yeah.
another oblong rock, which it looked
Another
like it appeared
oblong rock.
it had 6, 10 or more whelks on it.
Okay.
And again, the idea isn't to collect too many rocks, but instead of siphoning or slurping them up, maybe that's a good candidate
Okay.
to get whelks as well.
All right. Let's push in. Are you saving the slurps for something else?
Potentially, I just
Okay.
figured this
Hold
is
that.
a way to,
Let's back out a little bit.
yeah, save some time. And
Is it that one? Too much?
Rocks are good.
Keep going in. Right. Okay. Hold that.
yeah, now I think it's just to the right of the hockey puck, was it? I can't. It's below that octopus above the hockey puck. There's that oblong rock. It has some whelks on it. I think that was the one I saw.
Buddy, come on. We were having a discussion about how it used to be dredging for all the kinds of samples, but when you're doing it with an ROV, it's just much more personal.
Yeah. Yeah, think about some of those rock dredges. on seamounts just to get a few
Yeah,
samples and
yeah,
then think about what was in its path.
Yeah, absolutely, right? You're dropping a basket, towing it kilometers, saying the rocks came somewhere from in there, you think.
We call this a surgical strike.
Yeah.
Perfect.
Yeah, there's at least a
Oh,
Whoa.
crumbly.
Wow.
And I was about to say, you have to keep the tension just right so you don't crush the rock.
Yeah.
That'll happen.
Yeah.
All right, that's all right.
They don't count till they're in the box.
Right.
Rocks are quite altered, so they crumble.
Yeah.
Yeah, crumble.
There's one right in front of it.
What do you mean by altered,
Still
Rennie?
be good.
Well, thermally and chemically, not only from the vents, but just being in the ocean for however long they've existed.
So do we assume these are all volcanic in origin?
Yes,
Okay.
Mhm.
that would be my assumption.
So basalt, is that what we would guess?
Mhm.
Yeah.
That rock is bigger.
It's bigger, yeah.
But I'm neither a geologist nor geochemist, just a remote sensor.
You remotely sense that rock is bigger.
This
You-
time via camera.
You are the blind leading the blind.
Right.
All right, let's do that.
So you can check out the expedition tab of the website to learn all
Yeah.
about all the expeditions we have done this year. This is the 13th mission of the 2019 Nautilus expedition season and the final. So we've been all over the Central Pacific and the Eastern Pacific this year. Just a fantastic season of expeditions and highlights from those missions. So click around the expedition a little in the photos and videos tab to see some of what we've been up to. But this is a truly spectacular way to sort of wrap it up. Like, what a great thing that we were able to get back to the site where we discovered this a year ago, and continue to be gathering more data and learning more.
Ooh, that
crumbly.
is very crumbly.
Now it's just cradled, huh? So it looks like there's about 2 or 3 on there.
Mm-hmm.
It's good enough to collect, and we can get more whelks later.
Slurping.
Yeah, the slurp works really well on anything that fits in the tube and soft things that don't fit in the tube. Jamie, side box?
Yeah, maybe A since it's so crumbly.
Okay.
Starboard A.
Full wide video. All right, let's go ahead and switch our salvos. And open the box.
Again, if you're watching that quad view, great view of the samples going into the box and a view of the different divisions in our bio boxes. Hello to the STEM
Looks like
Foundations
those
class.
whelks are
who
at
are
least
tuned
one
in
or
watching.
2 are still there, so I didn't see any fall either. Everyone's favorite childhood game. Don't drop the whelks.
Oh, the hours we've all played that.
Lincoln Logs and whelks.
I've forgotten that one. Can you remind me how it goes?
Well, you have a spoon and some whelks. Run around the backyard.
Now the difference between the sample boxes are just size. They are divided and labeled. Again, very important as we're doing long dives like this where we hand over
Let
one
it
team
go.
to the next to have quality records. of where you're placing different samples, so we can correlate that back to the timestamp with video when we were there and all the sensor readings from that specific collection moment. So a lot goes into the labeling and just keeping the information straight on where we are and what we're doing. This dive is planned to be at 30 plus hours long, and we launched it last night about 11 p.m. local time, so coming up on 12 hours ago.
Sticky rock. Let's leave that open for now. Do you want another rock from that same spot with whelks on it?
Yeah, let's just look around for the smaller anemones that we saw.
Sorry.
Whelks.
No worries.
Any other
Does
conspicuous?
whelk have an H in it?
Yes.
Yeah.
Whelk.
Well,
Not like Lawrence Welk.
Quill, Cretan, Coo, Whip,
Whelk.
Well.
Quilk.
Video, let's push back in. Because I can probably get one of those other little rocks. Oh, there's a spot lobster.
Oh yeah.
If we want some more whelks.
Yeah, we do want some more whelks. I'd love to get probably 5 to 10 at minimum.
Okay.
Would be nice. We got like 2 or 3 right now.
Correct.
And if you think it's easier to slurp up loose ones there too, but
they'll come right off the rock with a little scrape. I won't go for these just because they're on smaller rocks and we don't want to send anything in. But there are plenty to get.
Like
Video, let's come wide.
I don't know if
Let's
those are within reach
hold
of the
that.
arm.
Yeah, for slurping. All right, video, let's push back in. You couldn't be on the other side. I mean, like, really.
I guess you can't really spin it around, huh? It's all tangled up in there.
So one of the objectives for later in the dive is surveying a larger area here, trying to understand the extent of these populations. We've already seen over 1,000 of these octopus, but we want to know if there are more of these seeps and more of these little pockets of it.
And lost the entire thing. Yeah, pretty
along
friable.
the way.
that's
So
okay. We'll have other opportunities if
Yep.
not here.
over the course
What
of the
else are we
next
looking
24
for?
hours or so, we'll certainly get to see more, or we hope we will see more.
We have anemones.
So let's keep an eye out for any smaller rocks that have those smaller translucent anemones on them.
Yeah.
Video, push in.
Ah, there's lots on there. That's amazing.
Mm-hmm.
Do you think that's a separate rock, Rennie?
Where?
I think that there might be a line right in there.
Yeah, it looks like that's in the foreground.
Yep. We can
Are
go
these
for that.
the anemones you're looking for?
That's a good question. Yeah, are these the anemones we're looking for?
They are the anemones. I don't know. It looks like the 2 females there have their eggs on a different rock. I don't know how much it would disturb them though, trying to get that one
Okay.
out of there, so we might want to avoid it for now.
Video full wide, we're gonna look around.
Oh, zoom back
Zoom
in
back
right
in.
there to that center, sorry. Right in between those 3.
Yep, you want that one?
And that's possible if the manip can get in there without.
Challenge.
It looks like the. It looks to me like the eggs are on other rocks, so
Yeah.
I think it's possible to get that one.
All right. Somebody needs to wake up.
Yeah, no. It was my
Full wide.
reminder to let people know that we're just coming up on 2 hours of pump time. So
Good.
we estimate that we have processed 72 liters of water for measurement of POP so far.
That's incredible.
Fantastic.
Come on. There we go.
Is that a filter system?
Yeah, well,
Aaron, let's push back in.
yeah,
Okay.
somewhat. It's there's a
Hold
particle
that.
filter
Mm-hmm.
in line, and then after the water passes through the particle filter, which captures everything associated with the particulate phase,
Heave.
the water then goes through a column packed with resin beads. That,
Resin beads.
yeah, which they have properties that attract nonpolar molecules in the water, so chlorinated persistent organic pollutants tend to stick to those resin beads.
Wow,
And
interesting.
yeah, so when we know optimum throughput so that we extract essentially all those POPs from the water before it exits the system, and then we bring the resin back to the lab. It is extracted with solvents and then dried down so that we get a very concentrated sample that then we measure the POPs.
Oh, there's eggs on
Oh,
it.
eggs on that one.
Okay, we'll leave it. We'll leave it then.
Yeah.
That's it. We've seen the mothers readjust and recover them. But look at that. There's only about 6 eggs and one that's probably has not hatched because I don't think the eggs are very developed. They look fairly opaque and white, so it could be one that.
Should we do a quick zoom?
Video
while
pushing
they're
all the way.
closed?
Maybe she's even gonna lay more.
Yeah, that one probably didn't hatch. It probably was consumed or something happened.
Well,
There's that little black spot. I don't think that's an eye spot though. It looks like.
Oh, but you know what?
It looks like she has more
She
eggs
has more
under
eggs on,
her.
yeah, on the
I
other
think
side.
those
On the
are
other
just
side?
some of them.
So maybe these are old rejected eggs.
that's incredible. There's
They've already moved
That
the rock
texture
since
looks
I touched
amazing.
it.
Yeah.
yeah, they sort of shoved it right away.
She seems to be protecting her other eggs in the same position, whereas these
Okay.
were kind of
Well-
off to the side.
I'm fine with collecting that rock.
Okay.
It's not 40 eggs. It's
Yeah.
5 or
Yeah.
6.
All right. Then we're gonna go back in.
Do you want me to go a bit wider? Are you happy?
Aaron, I'm always happy.
Oh,
You can hear the enthusiasm in my voice.
every day.
You're known for
Maybe
your
a hint.
enthusiasm.
Calm it down, Greg.
So while we collect this, a couple quick questions. These are Mesopteris
Yeah, she's got a
robustus, we
lot more.
believe.
Nice.
ROV Hercules is
Full wide.
built in 2001 and continues to be retrofit with different improvements and different types of samplers, which means it can do a lot of different types of seafloor operations based on what the needs of the science team are. So Hercules is turning 18 this year.
Yeah. We
Happy
can vote.
birthday. Yeah.
All right, Aaron.
Let's not go there.
All right.
Anemones.
Little
So
ones.
great.
Some eggs.
Little itty bitty anemones.
Wow.
Looks like really tiny tube worms.
Yeah.
You can see kind of their circular
Yep.
Oh, wow. Yeah.
calcareous shell there.
Yeah.
That's great.
That is. That's
Get
amazing.
a little micro community here.
All right.
Serpulid worms.
Jamie,
or
where do you
something.
want it?
B or C would be good.
B or C would be good. Full wide.
You said BRC, like bean, rice, and cheese burrito.
Don't make me hungry.
I know.
I was
Sounds
gonna
like
say,
time.
come on.
Yeah, a lot of times, I didn't skip breakfast today, but a lot of times I'll skip breakfast, and then from 11 till noon is tough. Especially Rennie's over here snacking.
And not sharing?
Never sharing.
I'm already stealing this from whoever left.
Yeah, I was
here
going to say,
last
Rennie
time.
is stealing, so
These are
it's-
He's
mine.
a scavenger.
You're just, yeah.
I can't offer
You're
what
just
isn't
snack cleanup.
Like the whelks of the
Yeah.
deep sea.
Welk is Rennie's middle name.
mine. Well,
Welk.
Welk.
There's
Renato Welk.
or not a well.
Herc, do you think that's too large or do you think it'll
no,
fit?
it'll fit.
Okay. So Jamie, we have 5th graders at Saunders Elementary from Virginia writing in, and they want to know what we do with the samples after we collect them, after we get them back. Can you talk a little bit about that?
That's a great question. Give me a minute to take a few notes from that sample we just collected.
No
And I'll
problem.
get back to you.
And are we looking at doing another sample real quick, or should we close the box?
Let's look around.
Okay, we'll look around first.
Jamie, can you confirm?
Yeah, you're not
11?
communicating, Chad.
Sorry about
It's
that.
okay, he's
Sometimes
loud.
forget. I'm
Yeah.
just yell across
Thank
the
you.
van here. Yeah,
Do
we'll
I
look
wanna
around for any other
come up a
conspicuous
meter or
samples.
two now that
Where are you?
closer to.
Yeah, we can do 28. The tether's been behaving, which is awesome.
So does
What's our
Nautilus
next
have the
target?
new?
Quick question. Hurt, can you confirm that that was B?
That was B.
Thank you.
Do we have the new cable management system on this year?
New cable management system.
Well, I thought they were going to redo things after last
Oh
year's.
yeah, there's a traction winch.
We have a
Oh,
traction
no.
winch, yeah, for our
Yeah.
main, for our main overboard cable.
You can
We used
see
to have a
that
direct drive winch.
Yeah.
monitor is the.
And it's all hidden down below. If you walk
Yeah.
down past the lab and noticed all that abundant new space.
Well, there's a bunch of boxes there now because we have the float and we have the shipping
Well,
crate
yeah.
for Atalanta. And
There's a
so
lot of
it's
space
been-
for us to stack up all the
it's
things
been
that we've
used
stacked
already.
up there.
Video push in.
Hey, Remy, out of curiosity, did you get nav points
That
for
one
our
is eating a
transects?
whelk.
I did indeed.
Okay, great.
Let me
Oh,
take
it's
a look at
eating-
the. Yeah, you can see
what?
the light green lines in the air.
Whoa!
So-
or being eaten
Yeah.
yeah, there
No,
they are.
she just
Eating
moved the whelk.
away.
No, to the mouth.
Yeah. So she's.
Well,
we need to
let's-
watch that, right?
Yeah, let's watch this. This is
Predation
new.
in action. Yeah, this is new.
This is new.
Does this count?
Go back to the arm. I'm going to use your camera to just
Wow.
kind of stow us a little bit better.
Okay.
I love watching the tube feet articulate like the little suction cups.
So
I
I
think it's
missed
cool.
the beginning of
Tube
this interaction. Did you see the
She
whelk actually
moved-
grabbed off
she
of the rock?
moved the whelk to the mouth. I don't know
So
if she
it
grabbed
was-
it off of the rock
Okay.
or not.
Okay.
So it might have fallen in place and then
Well, there's not a
Shuffled
lot of
down.
spot for it to fall from.
Yeah.
Wow.
Kind of an opportunistic feeding, maybe?
Well, she's certainly not trying to get rid of it.
No, if you go back in the video, you'll see that it was farther out on the arm and she placed it down.
Okay.
Okay.
Wow.
Wow.
Should I close the?
Just because we're zoomed in now, let's not bump anything. There's only rocks in there, so we're not worried about anything floating.
Can we listen for chewing noises?
Yeah.
It seems to be going
It
deeper.
is.
in
It
there.
is.
You don't want to be that
Wow.
woke right now.
No. No. Oh.
Well,
Well.
let's keep watching.
Well, maybe the whelk. Do these whelks sting? I wonder.
Yeah, I didn't see any proboscis
Yeah,
on them
right.
to
Okay.
deliver that harpoon injection like
Yep.
a cone snail.
I'm an assassin snail if you go freshwater.
Am I mistaken? Is that the opening towards us?
That is
Yeah.
the operculum. Yeah.
So
the
maybe
foot.
she's trying to figure out spinning around.
Yeah. Get the soft
Right.
soft pot first.
I've noticed that sometimes they will jet water at their neighbors. Just like, you know, they're jetting in one direction.
Oh, she
look
blew
at
it
that.
away.
She blew it away.
Huh.
That
Yeah.
is amazing.
That siphon came up and
Yeah.
over, and then
Yeah.
she gave it
Yeah.
one
Yeah.
large pulse.
Looks like she tasted it. Or
That seemed
That
intense.
was-
Not
was like
interested.
not
that was
interested.
not normal respiration. That was
Yeah.
So think about
directed behavior.
an invertebrate brain that has the
Wow.
coordination to control each sucker and then bring the siphon, the
The
excurrent siphon over to blow something off. That's just amazing.
she's
Yep,
cleaning
her
there's the
mouth
mouse.
or
Yeah,
something.
ooh,
Yeah.
get out of here.
It's like rubbing your tongue. Ew, ew, ew.
Yeah, that's right. Wow.
No, we're transferring human behavior.
Well, but I mean, no, it's coordination. It's like
That part,
there's,
yeah.
there's
Well, yeah, I
complex
couldn't do
behavior.
it.
That was awesome.
That was really
Well, to start with, I
cool.
don't have a siphon. So,
Yeah.
or suction.
Well, you wonder if there's some kind of chemical, obviously there's chemical sensing going on, but
Yep.
if there's something that was not tasteful.
Yeah, I
Yep.
wonder if volks are
Yep.
chemically defended. I don't know
Mhm.
that. Or how they are chemically
Or
defended.
maybe she just thought it was just the shell. Could be.
Mm-hmm.
But why would
Huh.
she have taken just the shell in the first place?
It's a good thought.
All right, what's
That's
next?
great. All right, so,
So
let's look around just a
interesting.
little bit here for any other
Tube
conspicuous
worms,
things we can
sponges.
collect. And if not, we'll wrap it up and start looking around.
And we're
this
happy
spa
on
for
our
a little bit
anemone
more.
collections.
Yeah,
Okay.
we only wanted one big one. We got
All right,
that.
video, let's push in. Let's hold that. Let's see.
Because we also have to balance out our load. I know we're going to do more collections later.
Yeah, and we have the plates. We've got plenty of ballast.
So Jamie, while we look around, could we circle back to the Saunders Elementary question? Of
Yes,
What do we do with the samples after we collect them?
Yeah, feel free to interrupt if you see something you want to look at or sample, but
Yeah.
otherwise, so there's some little sponges. Are you interested in those?
I'm going to allow or defer to Amanda for that.
Yeah.
I don't know which ones she may want to collect.
So once the vehicle is recovered back onto the ship, we take all the samples off, put them in little containers of cold seawater, and bring them back into our fridge. And then from there, we take them out one at a time and process them, which involves taking photos.
And are we going for either shrimp or spot lobsters?
Yeah, that squat lobster down there, that's a good idea. If you can slurp that and
Slurp
maybe
it.
some shrimp as well while we're here.
Okay, those go in different buckets though. Correct?
No, the shrimp and squat lobster, since they're all from the same location, could go in the same container. Or
Do you think they'll fight each
anything
other?
we...
Yeah.
Oh, unless you're worried about them eating each other.
That's up to you guys.
I'm not really worried about that.
I'm not worried about that.
Yeah,
Alright.
so anything we
Then I'm gonna
slurp from the same location can go in the same slurp jar, even if they're different organisms.
Okay.
Um,
I'm going to go ahead and close
so
our
while
bio
he
box.
gets positioned for that, I'll continue a little bit more, then I might have to stop to take more notes. But so then we take photos of each sample with a scale
Our
ruler,
numbers.
and then we preserve the sample in ethanol, which preserves the DNA in case genetic testing will be done later. And then we send the samples to the Museum of Comparative Zoology at the University of Harvard. And they have a large repository of samples there that any scientist can then access information about and request the sample to do more detailed analyses back in their own laboratories. And so that's for biological samples. For geological samples, we do the same procedure, except we just dry the rocks, and then we send them to the Graduate School of Oceanography at the University of Rhode Island. And again, any geologists or scientists can request those rocks to bring them back to their own labs and do tests of composition or anything else that they're interested in from that site.
Perfect. Thank you so much. So the slurp jars that you're going to get to see here, the slurp sampler is an underwater vacuum cleaner. It sucks up water and small things up through a tube that extends down off the right side of the porch. Goes up, sort of, you think about like if it's a person, you know, sort of up the shoulder, across the top, into slurp jars that are just above the light bar. So if this was your car, it would kind of come from the right side of the front bumper, up across the hood and up onto the top of the car in the windshield. So the slurp jars have different size mesh on those jars up at the top, and so they trap different things, different size things from escaping. So you can rotate like a carousel through the different slurp jars and decide on the size of mesh you want based on what you want to trap inside the slurp jar, and then turn on a vacuum cleaner that pulls water in along with small things like sediments or soft things that would be really hard to sample with the claw and put into a sample box. and then they get trapped up in the sample jar on the carousel on the top. The two laser dots are 10 centimeters apart. We use centimeters in science generally to measure, but if you prefer that in inches, it's about 3.9 inches.
Are we on the right? T2, 50%. Is it enabled? Yeah.
Should we jiggle the Geneva? Maybe it's not quite lined up.
Now it looks like there's some movement in there. Is there oh, back. Oh, okay. Which one? Yeah. Let me zoom in and that's as much zoom as I have. $50? Okay, we're at 50 now.
They're feisty.
Oh,
This one is not feisty.
moving up.
Not feisty, but evasive.
You're kind of pushing the rocks with the, oh no, you're good now.
Yeah.
So that's the slurp end of the suction sampler, looking to suck up some whelks right now, which have the curly shells.
We're actually after the squat lobster.
Oh, come here, squat lobster.
It's hiding behind the rock.
0?
Yeah, I don't think we're going to be able to get that guy.
Yeah, he might be exposed enough now. There, he's got some movement. Single arm.
Come on, squat lobster.
Yeah. No, it's not coming.
That's fine. If there's a shrimp locally or a couple of whelks we can put in. Oh, we have enough whelks. We don't need that.
We do shrimp.
Yeah, I can try a couple shrimp.
So, still, those rocks are kind of all we got so far.
0?
Nice, that'll. It's the only chance we got. Direct hit.
Success.
Well, maybe he's a magician.
Yeah. 1 second's there. Next, he's not.
We'll wait to see if it comes in the container.
Yeah, he's in
Yeah,
there.
and
there he
There
is.
he is. Yay.
Yay, we got him.
Success. Squat lobster.
Nice work.
Good job.
Hard.
Now I'm just thinking of B-52s.
12.
Yeah, canister one.
We don't have any units to measure how strong the suction is, right, Greg? I mean, I know we can dial it up from 0 to, oh, I can't hear you, sorry.
I don't remember what it was, but we did actually test it at one point to see how quickly it would move 5 gallons, and it was very quick.
Stronger than a shop vac?
Some good rocks.
Yeah, stronger
Okay.
than a shop vac. All right, Aaron, let's push back in. Let's hold that. Shrimp are hard. Okay. Shrimp will jet backwards, so the best way to get them is from behind. However, we're not in a great position for that, so we're going to try.
I was surprised how little that.
I think this shrimp is
Squat
actually
lobster.
farther back that way. Yeah.
Jamie, this is sample 12.
Yes.
Okay.
All right, go ahead and start suction.
To 100.
Yeah, let's do 100.
Ooh,
Oh,
yep, got him.
Oh,
Oh, no.
Oh, back up.
You want to 0 that, yeah.
and
get it
run
back
it
up.
again. Back to 100.
It was good work there to not suck up the octopus.
Good quick response.
All right, let's go ahead and 0 it.
See what we got in there. See a shrimp, at least one shrimp.
Yeah, there's a
Two.
shrimp in there. Okay, so let's go ahead and rotate back to flush.
That's great.
Okay, I see 2 shrimp and the squat lobster would still be in there.
Yeah.
Okay. And 1 hitchhiker.
So many good questions coming in. Thanks everyone who's tuned in and watching with us.
All right, Aaron, let's push in again. Do you want more shrimp or are you happy?
If we can get another one, that'd be great. And then since we're in the same location and we can double up on the cylinders.
Right, it can all go in the same cylinder.
Yeah, we can get more whelks with the slurp as well.
Okay. Yeah, I'm not gonna lie. I'm pretty sure a whelk went in during that whole process.
There's a lot of whelks up in the foreground.
There's a shrimp back there. That one's gonna be kind of hard to get.
Yeah.
So we have
Those guys are too far away.
a lot of questions that we're working to answer on these dives. You know,
That one's just teasing me.
things like how many total octopus are there out there. We're working on answering some of those.
All right, Aaron, let's come full log.
Where are these octopus when they're not brooding here? We have a lot of questions about that too.
All right, so we're gonna rotate back to 1 because we're gonna put it in 1 again. And you can start at 50%.
This looks like we're at 1.
Yep.
All right. You want me to start going?
Yeah, do 20% for now. It's just kind of to keep things from swimming out.
At 20.
Give it a bump in one direction or the other. Okay, there's flow. So we have some flow. All right, Aaron, let's push in. Let's hold that. You see what we're going for? Okay, let's go